So under screening of blood for infective material, we have a small test on a nucleic acid test. So this is a test now done by most of the modern laboratories. So it is if the test is done, the NAT testing is done on the donor blood, so it provides you additional safety to the blood donated. So it ensures greater safety in transfusion. So introduction of the NAT testing, it is a NAT, or it is also known as nucleic acid amplification test. So it detects particularly pathogen in the specimen. So specimen may be blood, tissue or the body fluid. So here the detection of pathogen is done, not the antibodies or the antigen. Widely used in many countries for the detection of HIV, hepatitis B virus and also hepatitis C virus. The method involves extraction of the genetic materials and amplification of the genetic material of the pathogen. So first it detects the virus and extracts the genetic materials and results in the amplification or the multiplication. So the detects the pathogen as soon as it is present. So no need of waiting for the development of antigen or antibodies. So it is a sensitive method. So most of the traditional techniques involve the presence of antibody to give the positive reaction, but NAT testing detects the viral genetic materials. If it is present in a lower level also, that is detected by this test. So one of the main advantage of this NAT testing is we can detect the pathogen during the window period also. So what is window period? It is the period between the onset of the infection and till the formation of antibody. So we can detect the pathogen during that period also. Since we are not detecting antibodies here, so directly we are detecting the pathogen. History. The method was developed in the year of 1999 and now it is widely used for screening HIV, HPV, hepatitis B virus and West Nile virus. Moving to the types, the NAT is depending on different methods. So the methods are based on PCR, branched DNA, ligase chain reaction, transcription mediated amplification, nucleic acid sequence based amplification. So these are the types based on methods. So now let us see method. So here, since we are talking about blood bank sample, so here the sample is our donor blood. So donor blood is collected in the blood bag. So blood bag, it may be the primary bag, satellites bag, and also we collect 5 ml of EDTA blood in the separate vacuum tube. So all these three should have a same ID. So the blood collected in the vacuum tube is sent for the NAT testing. Okay, so the sample here is 5 ml of EDTA blood of the donor sample is now sent for the NAT testing. So the procedure includes 4 important steps: sample pipetting, target capture, amplification and detection. So moving to the steps now. First step is sample pipetting. As I have told, the donor sample, that is EDTA sample, is sent. So here in this testing we can test 24 donor samples together. So 24 donor sample is tested here, that is not individually. Here 24 donors EDTA samples are there. No? So from there the separated plasma or the serum is pulled together in the single tube. So then that is tested. So here, so whatever the donor samples we have provided, so that is pipetted, that is only the plasma and serum part is pipetted to the separate single tube. So that is the sample pipetting. So that is done by the instrument itself, but it may take 3.5 hours. So sample pipetting, that is from the 24 individual EDTA samples, plasma or serum is pulled to the single tube in this step by the instrument itself. Target capture, now in the single tube we have all the pooled plasma or the serum. So from there nucleic acid isolation is done. So if the virus is present in that pooled specimen, so from that virus is detected and its nucleic acid is extracted or isolated. So this technique is done by repeated washing with the alcohol solution. So manually the procedure is done. So after isolation of the nucleic acid, now amplification is done. So here the genetic material is amplified or multiplied, or you can say single piece is made into bigger piece. So several copies of the genetic material is made by this amplification step. We have the separate instrument for amplification. So next, this many copies of genetic material detection is done by dual kinetic assay. We have other instrument for the detection purpose. If the genetic material is present here, the light emission results in the light emission that indicates the presence of virus. So these are the 4 steps of the assay procedure. So I will say here. So first, from the 24 donor sample, that is EDTA sample, plasma is transferred to a single tube. So in the single tube, so we do the alcohol washing many times for the extraction of the nucleic acid. So that is amplified in the separate instrument, so it makes more copies, and then it is detected by the dual kinetic assay and it provides results. Whether HIV, hepatitis B virus, hepatitis C virus is present. So this is the technique. We have the separate instrument for pipetting, amplification, and for dual kinetic assay. So this is the NAT instrument here. So that's all about the procedure. Observation: presence of virus indicates NAT reactive. So after that the sample may be tested repeatedly again, that is in the pooled specimen. If it gives positive, so we have to test individually now. So that is discriminatory test. We have to test for individual specimen. So few points. So if the pooling tests are done, it is done in the 24 donor EDTA sample. If negative means all the 24 samples present in the pool are negative. But if it is positive, what we have to do? We have to make a smaller pools, and then we have to make the individual test to identify which is that single donor unit is positive. So NAT superior over ELISA. So it provides additional safety to your blood. Instrument may cost of 60 lakh and the test may cost 2000 rupees. So benefits of this NAT testing: it detects even very low level of DNA or RNA present, provides additional layer of safety to the blood before transfusion, and have the ability to detect viral mutants. So it may result in the mutation of the genetic material, and any occult infection or hidden infection can be detected. The method is highly sensitive and specific for viral nucleic acid. It is faster than ELISA and it provides you better standard reports. So that's all about NAT technique.